Welcome to Cell Biology Lab. In today's lab session, you'll be learning about stomata movement in response to stimulus. So the aim of this experiment is to observe opening and closing of stomata in response to potassium ion concentration. So we know that stomata, which are small pores, which are evenly distributed on the epidermis of the leaves and also in the young stems, open and close in response to various environmental and internal signals. And this is regulated by changes in the trigger pressure within the guard cells that surround the stomatal pore. So in the coming slide, you can see how it looks. So here, this is the stomata, which consists of guard cells, kidney-shaped guard cells. So in today's experiment, we have kept two types of solution. One is hypotonic solution and the second one is hypertonic solution. So different concentration of hypertonic and hypotonic potassium chloride solution we have prepared for this experiment. And first we will see about what is the performance of the stomata when it is kept in hypotonic solution, lower concentration of KCl, where the concentration of KCl is lower outside the guard cells compared to the inside. So outside, the concentration of KCl is lower and inside it is less. So what happens? The water tends to move into the guard cells by osmosis because the solution outside the cells is more dilute. This influx of water causes the guard cells to swell and become turgid. As the guard cells swell, they become more curved and stomatal pore opens. So this is regulated by H plus ATPase, which is present in the plasma membrane that regulates the proton efflux from the cytoplasm, regulating the membrane hyperpolarization. So the concept here is hyperpolarization. Stomata opens when the stomata, if it is kept in hypotonic solution. The next case is hypertonic solution, higher concentration of KCl, where the concentration of KCl is higher outside the guard cells compared to inside. Water tends to move out of the guard cells by osmosis because the solution outside the cell is more concentrated. This loss of water against the guard cells to shrink and lose the trigger pressure. So as a result, what happens? The shrinkage happens and the stomata gets closed. As the guard cells lose trigger pressure, they become less curved and the stomatal pore closes. Therefore, in hypertonic KCl solution, the stomata are likely to close due to the decreased trigger pressure inside the guard cells. The membrane depolarization during the stomatal closure, it is regulated by anion channel activation and inhibition of H plus ATPase in the plasma membrane. So here inhibition and depolarization happens, and in the previous one hyperpolarization and H plus ATPase gets activated. So now you can watch the video how the experiment is performed and you can see the two stages, how the stomata gets open and when it is getting closed in the coming video.